Before I progress, and please let me know if you can see my screen now. Yes. Okay. So once again, I just want to say a big thank you to everyone at the CNPD. I've been more or less, sometimes I see myself as an affiliate of that center because of my long-standing relationship with Professor Saka, who is the pioneer director of the center. We've been collaborating for a very long time. And it's been one of the most productive, partnerships I have established, especially since I started to do research in the UK. So, right, I also want to say a big thank you for, inviting me to give this talk. It was a very timely invitation to give this lecture. As a matter of fact, this lecture is going to be the first kind of dissemination of our research on COVID-19 since we started. The work I'm presenting today has not been published. And on natural products, it's not been published. So it's an honor to do this this afternoon. So quickly, I just want to share some of the results, some of our results on African plants that we have investigated against this spike protein stimulated immune cells. So I'm going to provide a little bit of background as to how we got to where we are today. So on my screen, we are all very familiar with this structure. Sorry. We are familiar with this structure. That's the human coronavirus. That's the structure. That's the sectional structure. We can see that, the virus has some important features. And of all those important features, we are more interested in the spike protein, in the spike glycoprotein. And the reason we are interested in this spike glycoprotein is simply because one of the subunits, the S1 subunit, has been clearly demonstrated to be responsible not only for viral entry, but for immune reaction. So that's the component of the virus that is recognized by the body's, the host's immune cells. So, when we talk about something that a lot of people have been talking about since yesterday, that's the cytokine storm. By the way, before I started to attend this conference yesterday, I had this imposter syndrome because sometimes I go like, is the cytokine storm important or not? But from all the presentations I've had the opportunity to listen to in the course of this conference, I was like, yes, I think we're doing something right. So we are not the only people interested in the cytokine storm. So let me get back to the S1 subunit. That's the subunit that has been postulated to be responsible for the exaggerated inflammatory response by this virus. So, but that's not the whole story. Our research is actually informed by a few things. So we know that the spike glycoprotein binds to the host through the ACE2 receptors in the lungs. That has been established. We also know that viral replication and infection of other cells is one of the components of the pathogenesis with this virus. And we also know that the cytokine storm is responsible for a vicious cycle, which is actually what we are interested in. That vicious cycle that results in damage to the lungs, and not just the lungs, but other organs in the body. But at the moment, we are focusing on the lungs. We're trying to understand how the cytokine storm, the vicious cycle of the cytokine storm, is responsible for acute respiratory distress syndrome. Maybe later we will be focusing on other organs. So basically, at the moment, the strategies for treating COVID-19, of course, in prevention as well includes vaccines, the use of antivirals. Some studies have reported biologicals. Interestingly, many of these biologicals are aimed at inflammatory mediators. I'm coming to that later. And of course, anti-inflammatory drugs. And that's where our research comes in. At the beginning of the pandemic, I started to think about the cytokine storm especially. And even though at the time we weren't very sure about the exact role of the cytokine storm, especially in severe COVID-19, but I thought that, well, you know, it may appear as if there is an inflammatory component that we may target. And then subsequently, the recovery study was published, I think late 2020. And the recovery study, you know, came up with the fact that dexamethasone, which is a long-standing anti-inflammatory, provided some benefits. But of course, many of us would understand some of the demerits, some of the side effects with the use of dexamethasone. But that was one of the confirmations we needed when we initiated the study. So we started the studies, and then this recovery work was published. And that posed another problem for natural products research. And what was that problem? We wanted to investigate natural products for their effects or potentials in the cytokine storm, but there were no models. So that was a huge challenge at the beginning of our research. There were no cell-based models, there were no cellular models. All the papers, all the articles out there at the time were mainly describing or projecting or, you know, trying to explain that, oh, this natural product could be, you know, useful in COVID-19 because it's been shown to be anti-inflammatory in other studies, but that did not help us. We wanted something that mimics COVID-19 as much as possible. So there were studies published on, you know, natural products blocking lipopolysaccharide-induced inflammation. There were several studies on other instigators or other activators of inflammation, but that did not. We were interested in, you know, plants or natural products that would block inflammation or reduce inflammation in COVID-19. So our project at Huddersfield, first of all, focused on developing cellular models of COVID-19 cytokine storm, which we have achieved to some extent. And of course, subsequently, we started to investigate natural products which could reduce the cytokine storm and their mechanisms of action. Now, since June, when we started this project, when the project was initiated, we've investigated some plant extracts, mostly from Africa. And I'm sure African colleagues are going to be excited by some of the things I'm going to be sharing this afternoon. We also investigated some natural products from plants, and in the course of our collaboration, we started to collaborate with a colleague who is producing synthetic analogs of a natural product from parasitic worms. Interesting. The first thing we established, or we tried to establish, like I explained earlier, was to develop a model. So yes, I'm a natural products pharmacologist, but I needed a model to study my natural products, to investigate my natural products. So we did this study, and what we tried to do in this study was to look at the spike glycoprotein, the S1 subunit of the spike glycoprotein, and to determine whether, first of all, it could induce, cytokine production in PBMCs. And then we also had to find out, we had to investigate the concentration of this glycoprotein, the recombinant glycoprotein, of course, that could actually induce an increase in cytokine production. And interestingly, we saw that this glycoprotein was able to induce massive increase in the production of cytokines from these cells. And we validated our observation with of course dexamethasone, which was the mainstay at the time for, and I think it's still the mainstay for the for the treatment, as part of the treatment strategies for COVID-19. So this is an article that was recently published in Inflammation. So after we had this, we were ready to progress with our studies on natural products. As a matter of fact, when this work was published, I told colleagues, I said, no, this is not where we were going. This was just to establish our cellular model. And, that has worked fantastically. So going back to natural products, the very first, substance, if you like, that I was interested in was Garcinia cola for several reasons. This is a seed that that has been part of my life since I was young for several reasons. I do not have time to go into that. In addition to that, when COVID started, especially in Nigeria, I'm originally from Nigeria, people were talking about different things that they've been trying. As a matter of fact, earlier today on the chat, some people mentioned certain things that were being used in Nigeria, and we have investigated some of these things. So we were interested in Garcinia cola in the first instance because of what I know about it, anecdotal evidence of what I know about it. And in addition to that, several people have investigated some bioflavonoids from Garcinia cola, but this time I thought, let's look at something else in Garcinia cola, because those biflavonoids, as a matter of fact, I think I had this chat with Professor Saka at some point. They are still, those things are still like extracts, but we needed to find out what could be, responsible, what else could be responsible for the activities that have been reported about Garcinia cola. And this vitamin E analog that has been shown to be one of the major components of Garcinia cola came to mind. It's garcinoic acid. So we decided to investigate this too. And what we did was to, to have PBMCs. These PBMCs were pre-treated with either Garcinia cola extract at those concentrations you can see on my slide. And we standardized, we controlled the experiment using 100 nanomolar of dexamethasone. Why did we use that concentration of dexamethasone? That was a concentration that was shown to have optimal anti-inflammatory activity against spike protein induced exaggerated inflammation in our previous study. So we adopted that concentration, and I'm not going to spend too much time on this. We can see the concentration dependent anti-inflammatory activity of Garcinia cola extract in this study. The levels of TNF-alpha, IL-6, IL-1 beta, IL-8 were reduced in the presence of Garcinia cola. In addition to that, garcinoic acid had a similar, with between 1.25 and 5, we actually discovered that beyond 5 micromolar of this compound, there was cytotoxicity. But between 1.25 and 2.5, we saw similar profile, similar pharmacological profile against inflammation, increased production of pro-inflammatory cytokines by the spike protein. And one more thing I need to mention here. So we used 100 nanograms per ml of the spike protein to induce exaggerated production of inflammatory cytokines. That was the concentration that gave us the optimal increased production of this pro-inflammatory cytokines. So having established that these two, the extract and glycinic acid, were able to reduce, increased production of inflammatory mediators, we also were interested, to some extent, in the mechanism of action. NF-kappa B is a transcription factor that is known to actually control, gene expression of many pro-inflammatory cytokines. And also in our study, in our modeling study, we saw that the spike glycoprotein actually activated NF-kappa B, massive activation of NF-kappa B, which explained, the role of the spike protein in increasing the levels of pro-inflammatory cytokines. And we also, in these experiments, we decided to add BAY11708-2, which is a known NF-kappa B inhibitor. And 2 experiments were done. One experiment was to measure the DNA binding capacity of NF-kappa B, and all these were reduced in the presence of Garcinia cola extract, dexamethasone, and BAY11708-2. In addition to that, we carried out a reporter gene assay, and the similar trend was observed. And in this assay, we can see that there was an increase in the activation of NF-kappa B, transcriptional activation of NF-kappa B, and these were blocked by Garcinia cola. A similar thing was observed with glycinic acid. This confirmed our earlier observations as to the potential of Garcinia cola on the one hand and glycinic acid on the other hand in reducing inflammation, increased inflammation or hyperinflammation induced by the COVID-19 spike protein. Okay, we didn't stop there because we had an aim, and that is lung damage. So we went on to do what we call a co-culture experiment. So we cultured PBMCs and lung epithelial cells. We cultured them together, and again, we stimulated the PBMCs with the spike protein. And of course, after stimulation with spike protein, we brought the PBMCs in contact as much as possible in a transwell with A549 cells. And you can see that in the presence of the spike protein, there was a massive release of LDH, which is an indicator of cell death. In the presence of the lowest concentration of Garcinia cola, there was no significant reduction in cell death, in the death of the lung cells. But with 12.5 and 25 micrograms per ml of this extract, there was a significant reduction.
2 minutes left.
Same thing happened with glycinic acid. There was a prevention in damage to the lung cells co-cultured with PBMCs. So those were some of our results. Of course, there are more, but I do not have time to share all of them. We've investigated extracts and alkaloids from Xanthoxyloides. Professor Saka knows more about that. Hopefully, we'll be talking about this some other time. Same thing with Azadirachta indica. These are the other natural products from African plants that we have investigated. So what do we currently know? First of all, we know that the spike protein plays a major role in COVID-19 cytokine storm. We also know that, this excessive release of pro-inflammatory cytokines is a potential pharmacological target. We also know that some natural products from African plants, they show some promise in SARS-CoV2 cytokine storm. So these are the things we are trying to establish. We are trying to understand better the inflammatory signaling cascades in macrophages. We want to know, I do not have time to talk about the toll-like receptors and ACE2 at the moment, but we want to know more about how the spike protein actually causes inflammation in these cells. Is it through TLR4 or ACE2 or both of them? And we are also interested in further establishing the roles of these signaling pathways, NF-kappa B, the p38 MAP kinase, the NLRP3 inflammasome. At the moment also, we are thinking about investigating natural products for the so-called neuro-COVID-19. We published a paper showing that this spike protein is also able to induce neuroinflammation in BV2 microglia. So these are the people who supported me with this research, and two of them, they were all PhD students. One of them has graduated now, he's now a postdoc in Poland, and two of them are still their current PhD students. And these are people, I mean, organizations, the FRIN in Nigeria, University of Strathclyde, OAU in Nigeria, and of course, the Center for Natural Products Research. I hope to be able to explain or to present some of the results we got from natural products that were prepared and isolated from this center. And of course, we've got an existing collaboration with PhytoQuest. So we are inviting you to join us if you're interested in collaborating with us. We've got a database in the Academy of Medical Sciences Preclinical Drug Development database. And this database explains the research that we do. Thank you very much for your attention, and thank you very much for your interest in this talk.